controlled on this computer. Done. Let's go back to the fluorescence. There. So these resources for fluorescence and filters about spectra and things that are very important to check before. On the microscope, in the real life, not a simple version of a microscope, it looks like that. This is the same microscope that is in biochemistry. It's for the fluorescent. Most of the items are motorized, so you control them with the computer. And there is things that I didn't talk about, but usually when you get a training on a specific microscope, I explain what it's used for. And I put these little hands when something is manual, so you cannot control it with the computer. So yeah, that's the example. Fluorescence microscopy, why is it useful and why you should use it? First, you can work on, it's very convenient because you can do a fixed tissue or live tissue. It's very easy to use. You can do both transmitted light and fluorescence on the same wide field microscope. And it works for short images, but also time lapse and things that are very long to image. The benefit of the fluorescence and the wide field is that it's very easy and versatile technique. But one of my favorite ones because it's not fancy and it's almost work all the time. So it's very approachable way and it's not very expensive. So yeah, a lot of advantages. It works better with thin samples, but you can also look at thick samples, but it's not meant for that. So there is a drawbacks. And usually what you can see in fluorescence on the wide field microscope on the camera, you can also see within the oculars. This is not true with other techniques that we will discuss later. So this is one of the main advantage. People like it because it's real. The drawbacks are bleaching. I won't have a dedicated part on bleaching, but this is a very important component. It's just we lack of time. The axial resolution is limited. You cannot do multicolor on the wide field microscope at the same time unless you have different detectors. And this is possible, but it requires having different detectors. And also there is a lot of blurriness because there is some light out of focus. The resolution in fluorescence microscopy, you see you don't have a condenser anymore. So the Rayleigh criteria tells you that the resolution you can get is 0.61 times the wavelength divided by the objective numerical aperture. And the axial resolution is this. So if you look at objective with a NA of 1.4, and if you look at GFP emission, which is 510 nanometers at the maximum peak, it gives you a lateral resolution of 220 and an axial resolution of 1 micron, 1000 nanometers. Just as a reminder, for transmitted light, you have to add the numerical aperture of both objective and condenser. That's how we end up with the wide field microscopy. So we did wide field microscopy, transmitted light with a lot of different contrast method and fluorescence. So this is a very important part to give you really the basic of the microscopes. And then now we're going to go into more details. There is a lot of things I didn't talk about. I didn't talk about light sources. There's different kind of them, and they have different properties. But this I prefer not to describe it during the talk, but more into a instrument basis. There is different technique aspects for staining your samples. You can use colorants. You can use proteins. You can do things that are indirect or things that are direct. And there is also that this kind of positive and negative control to be used. I didn't discuss that. I also didn't discuss the fluorophore, the different kinds, and they have different properties. But I didn't go into these details because I want to focus on more practical aspects that are related to the microscopy. So just to come back to the initial methods, we just look at the wide field. It took a long time to go through this little part, but the good news is going to be much faster for the rest because you know all the basic, and the rest is going to be just one single technique. There is no contrast method within these techniques, so don't get scared. We're going to finish within time. So the first one that most of you guys might be interested in is confocal point scanning microscopy, and I'm just going to give you an example of why do we use that. You know that if I image a point, I get a blurry image. This is called the point spread function. This is in X and Y, so you look flat at your sample. This is X and Z, so you look vertical at your sample. So if you have a point object, you get a blurry dot. And this is because there is a lot of signal that is coming out of focus. So is there a way to kind of bypass that? Yes, there is a way. Very simple way. If you have a point and you put a pinhole within the two lenses, you can actually block the light that is out of focus if you put the pinhole at the right point and the right size. So in this case, you see that you have your focal plane. The light from this focal plane that is here is going to the objective, but the light that is outside the focal plane is kind of blocked by this pinhole that is here. Again, here it's out of focus and it's blocked. But when it's focused, then it gets through the pinhole and it gets to the detector. So that's great. We kind of remove all the light that is outside of the focal plane. It's not perfect. We still have a light that comes from outside, but it's more limited. But the problem is that it works for only one single point. And usually when you look at a sample, you don't look at one point, you look at many points at the same time. And that's where the video from confocal microscopy comes in. Didn't read the video. Oh yeah, we did. So in a confocal microscope, this was the example of the fluorescent microscope that we saw before. And now we are putting a pinhole right there. In this case, they put two pinholes, one here. And this is actually focusing the light into a single spot within your sample, a very tiny spot. Then the sample is going to emit some light. This light is going to go back to the objectives. It's going to cross the dichroic, go through the filter, and go through another pinhole to get detected. So if we do that, then we have a single point from the sample. The problem is that the sample is much bigger. So we need to scan the sample point by point. We have to move the sample or move the ray of light. And if you do that, you can actually scan your sample. You can do it in an X and Y, but you can also do it in a Z region. So in this example, he moved the sample a bit up and it went down with the light, and now he's scanning the second part. And if you repeat these steps, you can actually reconstruct a 3D volume of your sample. In the confocal microscopy, we don't move the sample. We usually find it much easier to move the light instead of moving the sample. And that's the result what you get. You don't get a single image, but you get a 3D volume of the image.
How frequent is the use of an emission pinhole like this in standard confocal
scanning? In the standard one, I don't think they use it anymore because we have much better way to select the light. It's still done in a spinning disc confocal. You have a pinhole at the beginning and at the end. Yeah, it's not something standard. I think it's because the people that made the video are really into physics, but they use this. So it was not about only optical microscopy. So point scanning confocal, you have to scan the sample in X and Y, and if you want to, you can also scan in Z. There is also consequences. You cannot use any kind of light. You need a kind of a stronger light, and we usually use lasers because laser, they give you one single color. It goes into one single direction, and it's spatially coherent. So light is in phase, and it's most of the time also polarized. So we use this laser. And also because we are looking at one single point, we don't need a camera. Camera has many pixels. In this case, we are using kind of a camera that has one single pixel. It's called a photomultiplier tube. And the principle of these guys is that they can amplify the signal, and they are very, very, very fast. So if you don't want to spend too much time for each point, you just use a fast detector. And I'm just going to give you a bit of an example of what is a photomultiplier. It's a little tube. Physically, it looks like a tube. It looks like a single tube like that. The light goes through either from the top, either from the side. And you have a photon that comes in, and this has a specific media inside or material. When a photon comes in, it can kick out sometimes an electron. And this has a specific efficiency. It's not all the photons that's going to give you an electron. If the electron is emitted, then it can be amplified by a series of diodes, electrodes. And at the end, you have an anode that is going to measure the current that you get. So you get a photon, you convert it into an electron, you get it amplified, and you measure what is the voltage that you get at the end. The downside part is that here, the efficiency between the photon and the electron, it's not as good as cameras. So the best of this guy can be 60 or 50% efficiency. So you get 2 photons, but you can measure only 1. So you lose a lot of light, but you can do this in microseconds. What does it look like, and why you can use it? Well, you can look at the live sample or fixed samples. Most of the time, the live samples, there is a compromise between the speed of the scan and the imaging time. You can look at the transmitted light, but also fluorescence, and mainly fluorescent. And the main advantages is you have a very high resolution. And this is the 2 examples we have at the Faculty of Medicine, and this is an example of the cells you can get. You get rid of all the light that is out of focus, so you can have a very crisp and sharp image. And people like it a lot because it gives you stunning images to put in your publication. So the main advantages are what? Very good resolution. So we are actually reaching 200 nanometers and 500 nanometers in Z. You see that you have a big increase in the Z resolution because we remove a lot of light. So it gives a great and crispy images. The good thing is how it's built. You can actually change the field of view, and you can scan a small region if you are interested in a small region, or a big region if you're interested in a big one. You can also put a lot of detectors at the same time, and you can detect several colors at the same time, so which is very good because you're going to save a lot of time since you have to scan the sample. The resolution can also be changed. Instead of doing 1,000 points per line, you can choose to do only 500 or 256. So you can adapt the resolution to what you actually need. It works very well for thicker samples, so you don't have to be very, very thin because you remove the light that is out of focus, and you can do 3D reconstruction. The downside is it actually is very slow. You have to scan each line and each point individually, so it can be time consuming. You also bleach a lot because you put lasers and you focus the light on a very small spot. You bleach a lot of your sample, so you can do 2 or 3 acquisition, and sometimes the sample is dim, so you cannot acquire anymore. And then one of the downsides also, it's more complex to use and to find the focus, because you don't see the light that is outside the focal plane. It's kind of hard to find the focus. I just want to show you an example I find what is done with confocal microscopy on both transmitted light and fluorescence. This is a cell that is going to divide, and you see that it takes several hours for the cell to divide. So in this case, it took 4 hours for the cell to divide, but you can actually measure that. So it's not so good for things that are very, very fast, but things that are slow, such as cell division, this is a good tool to use. So we saw wide field microscope, we saw point scanning. Now we're going to go to a different kind of point scanning. It's called confocal, spinning disk confocal microscopy. The main concept is, instead of using 1 point at the time, if I put 2 points and they are separated by a good distance, I can actually scan 2 points at a time without, a big trouble. If I put a lot of them, then and they are far enough, then I can actually scan each point individually. But I cannot use a single pixel detector. I need a camera this time, but it has to be a very sensitive camera. So I found this video from the company Yokogawa that explained to you what is a wide field microscope. So you have the light, it shines on the sample, and the sample is going to emit light, and you can detect it. But because there is no confocal plane, it's kind of blurry. It's a company, so they make things exaggerated. It's not super, super blurry like you see. It's a bit blurry. So then if you want to do a point scanning microscope, it looks like that. You scan with your laser through your sample. So you have a sharp image, but it takes a bit of time to kind of scan it line per line. And that's what you get. And also you have photo bleaching, so the light is kind of diminished. Then they made this kind of single spinning disc microscopes where you have a disc which is punch of hole and it's rotating, and the light goes through that, so you can actually screen your sample with many different points. And this gives you a kind of a spinning disc microscope technique where the ray of light are scanning very fast the sample, and then you can get a confocal image at a very fast speed. So just a bit of a detail about the micro lens that are on the spinning disc. If you shine the light on the holes, most of the light is going to hit the side. But if you put micro lens, then you can converge this light and have more light going through each hole. And that's this Japanese company Yokogawa that patented this micro lenses, and that's why it's so good. So we have one in the biochemistry department, and this is the most heavily used microscope because it's fast and it answers a lot of application. So this microscope requires a camera, and camera are different from the photo multiplier I showed you before, but the principle is the same. You want to start with photons, and you want to convert directly to a signal that your computer can measure. This is done here. At the sensor level, you have a photon hitting the sensor, and it's going to convert into an electron. But this is not perfect. Sometimes you need several photons to convert them into one single electron. The best cameras have a 95% quantum efficiency. So if you have 95 photons, 100 photons hitting the sensor, you have 95 electrons, which is pretty good. But older cameras have like a 30% efficiency. Then these electrons sometimes get amplified by a gain, and it get also digitized by analogic to the digitizer, and then you have your gray levels in the computer. And if you look actually in the side, you can see that sometimes they put micro lenses on the top of the pixel. So the pixel is here, it's very small, and then they put a micro lens to converge the light in there. Different kind of cameras, they have different properties. I think for you is not too much of matter. You can rely on our expertise if you want to ask questions about cameras. But for time constraint, I'm just gonna jump into this camera session. Oh yeah, an important thing is I show you the cut of the front TMOS, where the light is going front a micro lens and go through all the wiring and get to the diode. They also have this back illuminated where they actually just reversed everything, so the light comes from the back, and this is much more efficient. One of the important things about the camera is the quantum efficiency. This is a value that doesn't get bad. You can rely, if you want to buy a camera, this is almost the only thing that matters, and also the resolution of the camera. This efficiency depends on the wavelength, and you see that, for example, the early CMOS that are here, they have a not so good efficiency into the infrared, so you cannot see your Psi5 or Psi7, but we are pretty good here. We are 60%. Now we are reaching 90% at the peak, which is between the green and the red. Another parameter important is the size of the pixel. This influences both the resolution and the sensitivity. It really, a pixel, we can really think as a bucket, and photons are running into this bucket. So if you have a big bucket, you get a lot of photons, and your pixel is going to be very sensitive. If you have a small one, it's less sensitive. But the size of the pixel is also important for the resolution. You take this example of this camera, which has big pixel here, and you have this object you want to image. I have only 4 pixels. And if I reproduce the image of these 4 pixels that get illuminated, I got the square. But my real object was two bars, so this is different. If you have a camera with higher or smaller pixels, then you see that you can actually resolve better the object. So the size of the pixels is important for the sensitivity, but also for the resolution. Also the speed, some people want to image things that are very, very fast in a cell. I'm going to show you example later on. So the speed of the camera is a very important thing. You can always decrease the size of the sensor digitally to kind of go faster, but usually people want to have the full view. So in the spinning disk side of the speed of the camera, the speed of the disk is also important. If you have a mismatch between the speed of the camera and the disk, then you want to actually see the ray of light because you are not doing a full cycle around the time you have to image. So you have to match the speed of the spinning disk and the speed of the camera. An important thing about an important characteristic about the camera is also the depth of the well. So if a pixel is a bucket getting water and it's very, very deep, then you can put a lot of water and you can still measure how much water you have. If you have a 1 liter bucket, then you're going to just be able to measure between 0 liter and 1 liter. But you have a 10,000 liter bucket, you can measure a lot of difference in terms of volumes. So you can really think about the pixel as a bucket, and the depth is important because the higher depth, the higher details about the brightness you can measure. Another thing important is about the shutter. Some cameras have a global shutter, which is all pixels work at the same time. Very simple, like a regular digital camera. Some other camera have a rolling shutter, and this has a benefit sometimes. But if you look at things that are very fast, because the pixels are not illuminated at the same time, it can cause trouble, and there is a deficiency. This is an example of a spinning disk confocal microscope. You see the microscope, the spinning disk is right there, and you have a full laser module on the side. It's very good for live sample. You can look at transmitted light and fluorescence. And if you look at fluorescence, it's going to be confocal. And it's designed for live time imaging and time-lapse imaging. And just going to show you a video from cells that are expressing single molecules in fluorescence, and you can image them over, I think, 5 or 10 seconds. So this is very fast happening in the cell. There is almost no bleaching into this kind of sample. Here it's 15 seconds. You can see a bit of the bleaching from the end to the beginning, but it is really limited, and you can still see your cells. And remember that you don't need to have a pretty picture to get result. In this case, these people are interested into the movement of each of these particles, so we're going to track it into a very short period of time, and that's all what matters. It's the numbers. It's not really how much the resolution of the image is. Another example of a spinning disk confocal microscope is you can look at cells dividing, and this one have a label that show the cell phase. So there is G1, G2, and division. So they change color over time if you look at them when they divide or when they are in a specific phase. And when they are in, I think, mitotic cells, they just lose all the colors. So this is something you can do. This is, I think, 12 hours videos or several hours video. And to show you what the company sells on their website, they can actually also look at more complex behavior of neurons or cells over or another layer of cells. Confocal spinning disk confocal are very good too. So the benefits, very fast, almost no photo bleaching, especially for long time lapse. You can do a multicolor if you have multiple camera, and you have a very good axial resolution. Lateral resolution is limited because of the camera, and finding the focus can be difficult. So now we're going to move to super resolution microscopy. And this is very interesting, and a lot of people are showing interest and comes to me with questions. And what is super resolution and why do we use it? I told you that if you have a single point and you image it, you have this kind of airy disk. And this is, it doesn't matter the size of the point you are using, it's going to be that size of airy disk. So any technique that is able to go smaller than this airy disk is a super resolution technique. The first one I'm going to talk about is called Palm or Storm. I'm just going to go briefly. because it's not heavily used. And I'm just want to present you so you know what we can do that. So how can we refine a smaller airy disk? Well, one of the things, instead of imagining all the pixels at the same time, all the fluorophore at the same time, let's imagine I just eliminate a few of them, and these guys are kind of far away. If I eliminate one single fluorophore, I'm able to kind of retrace, knowing that I'm going to image this with an airy disk. I can say, okay, I can see all of that, but because I know that I have only one single one, I know that it's right there. And now I do another image where I image another single molecule. And I say, okay, another one is right there. And then you have this kind of image that is reconstructed. So Palm and Storm are two techniques that use different methods to kind of image fluorophore one after the other. So instead of eliminating all of them, we're going to split this in time. So it obviously takes a lot of time. You sometimes need to do 10,000 images to get only one super resolution image. So that's the drawback. But in terms of resolution, you get things that are much higher resolution. So here you have a confocal image microscope, which is a good resolution. And for some people, it's going to be enough. But then you have the Palmer or Storm resolution, which is much greater. Another technique is called expansion microscopy. It's not really a microscopy technique because it doesn't use light to have a better resolution of the sample, but instead it's just making the sample bigger. So what you're going to do is you take your sample, you're going to put a mesh around, but a flexible mesh, and it's kind of a gelatin if you want, and then you're going to dip it into a water or another component that's going to kind of blow up. So it's going to take up the water, it's going to blow up your sample. So at the end, your sample is bigger. It keeps the structure, but it's just bigger. And then if you go with a regular microscope, you can actually see with better resolution because things move away from each other. So I included it into super resolution, but it's not per se microscopy technique. It's more like a way to get a better resolution, but not a super resolution technique. I think at Saint Justine they are very interested to kind of to try it at least. I would be very keen to try it with something, but this requires more lab work than microscopy work. And because I don't have a lab where I can do wet bench, that would be cool to kind of try this kind of thing. Another super resolution technique is about spinning disks. So we come back to the spinning disk. If you put micro lenses here at the bottom part where the light gets excited, where the light gets emitted, then you can reassign this light to a smaller point. So you get more light, and this increase the resolution of spinning disk. This is called super resolution spinning disk. I didn't see it in effect, so I never tried it. I just look at the data online, and it looks pretty good. I don't know if you can see the details, but this is a blurry image of the white field. This is with deconvolution. This is the spinning disk super resolution, and you can also do deconvolution on that, and that's the result that you get. So it's crisper and sharper image. Another technique is called STED. It's really interesting technique because you want to eliminate a small point and get rid of the blurriness around. And how they do that instead is they use two lasers, the excitation laser, like a regular confocal, but around this laser that is here, you're going to put another laser that looks like a donut. And this is going to deplete all the laser light around. And because with light, we can actually play very well with the intensities and the position. If you combine both, you have a very, very small point to excite light. And knowing that you can actually reduce the excitation place, then you can have a better resolution. And this is the truly super resolution technique that kind of changed the how we were thinking about microscopy. So here again, an example of confocal microscopy images, and that is a STED image. Another thing about my super resolution microscopy, it's called the structured illumination microscopy. So what we do here is we do a pattern of illumination. And by combining several patterns, we can have this kind of interference band. And same principle, but instead we're gonna narrow down the place that is actually illuminated. And if you do that, and the good thing is we have one such of the microscope at the Faculty of Medicine, you can look whatever samples you have into a super resolution technique. You can look at live samples, but if they cannot move too much because it takes a bit of time to take images, you can look at transmitted light and fluorescent, and you have all of this in super resolution. So the main advantages is that you have a very good resolution. So you can go down to 100 nanometers or 150 nanometers for the X and Y, and down to 300 nanometers for the Z. It's very easy to use. You can just instead of taking one image, you take 15 images and you calculate the super resolution after that. And you don't need to have specific labeling. The limitation is that it's a bit slow. You have to take at least 15 images to get one images. You have to use a very strong power, so there's a lot of photo bleaching. You cannot do a multicolor at the same time. You have to use the same camera several times, so multicolor is a bit even slower. There is some bleed through between channels, and you have to do post-acquisition processing. There is also artifacts that come from the principle of super resolution. If you have a very good sample, then you don't see this artifact. That's the end of super resolution. So there is other techniques that I didn't talk about, and I'm going to go very fast at the end because I want to focus onto the last part of the talk. So turf is a technique where you don't shine light directly to your sample, but you give it an angle, and it just it's reflected back to the objective. And by doing that, you can actually excite a very, very, very small, narrow places, and this gives you very sharp images. So turf is very useful. Multiphotonic, instead of using one photon to excite your fluorophore, you're going to use a combination of 2 or 3 photons to excite. But these photons have to arrive at the sample at exactly the same time. And if you do that, instead of having a cone of light that is very thick such as a confocal microscopy, you get a very narrow excitation point. And if you look at that in real life, you get this. This is a confocal microscope, so you have a very big cone of light. This is a 2-photon microscope where you activate the light very, very in a very small point. Multiphotonic is very good for in vivo because you don't photo bleach, but you have to scan. And this is an example of T cells growing into a tissue that I don't know of because I didn't find the references into this video. So you can actually image this for a long time, and there is no photo bleaching. All right. Any question? I guess not.
In terms of super resolution, so there's a sim chemistry platform.
Yes.
Are there any D-Storm or STETS tools?
Yes, we have several of them. The SIM is able to do D-Storm. Any kind of epifluorescent microscope can do storm and palm. But you just need to prepare your sample in a specific way, and you have to process the data in another way. So, and the limitation is also you need to have a very fast system, because since you have imaging for palm and storm for a long period of time, if you are not able to take 1,000 images a second or 100 images a second, it's going to take too long to image. So yeah, I think we are into the mood of bringing more super resolution microscope. But yeah, funding is an issue. And for the STED, there is no plan to get one real soon, but there is one in Saint Justine that is open and usable, and I think they have one at Eric, so not too far from here. Last part is about acquisition and really the tips that help you, and the practical guide for acquiring image. The first thing I want you to know is you need to know about your microscope. And if you don't know which microscope to use, come to us. We know, and we will help you to find the best tool you need. We actually made a website, wiki, that I want you to see there. So you go to wikimontreal.ca, you search for microscopy FacMed, and you have this website. What is good about with this website is that we are several people in charge of maintaining it, and we update it with a lot of information. The first thing I want you guys to do is actually connect yourself to the login page. If you are at UDM, then you have a login page. I have this issue from the IT today, so I cannot log in because I changed my password 2 days ago. So I have to fix this issue. But if you go back to, but you will be able to log in. If you log in, you are able to follow a page by clicking on a watching button, and you're gonna receive notification if I change the page or if I do something. So you can stay aware of what's happening to a microscope. I put the microscope that are available currently. There is a super resolution microscope, there is a confocal microscope. And if you want to actually find them, you just go to instruments. You can go in English if you want, and you can click what do you want to do. If I want to do point scanning, I click point scanning, and this is the microscope that is available for point scanning. Okay, click on it, and then I have the information about it. If I go to another microscope such as this one, this is a upright microscope. This is another this is an inverted microscope. And you can find the user guide, how to use it. You can also find the light path that tells you everything you need to know about the light path. There is the manuals from the company, but I don't find them useful for you guys. I also put a log. So if the microscope doesn't work, or if it's something that I've done on it, you are aware of what's going on. And this is very important to keep user aware of what the machine is doing. There is also technical data sheet. But the most important thing for you is the description. I told you about the specification of the objective, and everything is there. There is also the complete specification about the light source and the complete specification about the filters. Why I put it there and why I don't refer you to the website? Because the company doesn't provide all the information all the time. For example, recently I bought an objective from a company and I asked, oh, can I have the transmission curve from the objective? And they told me, oh, no, we don't give you this information. So I was really pissed off. So what I do is anytime I have this information, I put it on the website. So this is the objective from Zeiss, but I also put the transmittance, which is the most important thing into the objective. So here, if you click on that, you have the curve of the transmittance of one given objective. So yeah, the company doesn't play sometimes fair, but I keep all the data relative to the instrument here. You also, there is the contacts, you can contact us. So the wiki is a very good source. Anyone with a UNIP will have access to that. Yep, question. All the microscopes are in the biosafety level 1 room, but you can use bio level 2 as long as the sample is confined. We don't go into 3 because 3 requires any kind of ventilation. But as long as your sample is sealed and that you transport it to the microscope in a sealed fashion, then it's safe. We also need a protocol that if you spill something, you know what to do. So level 1, level 2 biosafety, this is taken care by ourselves, and we have protocols. But you can use a level 2 into a level 1 room as long as it's confined. So what I really want you to do is to click on the wiki, use your identification to log in, and you can follow the page. You can have all the technical information. It's very important. The second thing about after the instrument, it's the biology. You need to know what is your samples made of and what do you want to do in your experiment. So for example, the size and the resolution that you need. Do I want to see a full cell or do I want to see a mitochondria? So this is a question you need to know before doing your experiment. You also need to know what fluorophore should you use or what fluorophore you are using. Very important questions to check that the fluorophore are matching the filters, for example. You have to be aware of the photoblanchiment and the phototoxicity. Two important things that I put on the blog of the wiki. The wiki also have a blog. And I can refer to that, but I don't have the time to talk about it. You also have to know what speed you want to image. Do you want to look at something that is fixed or you want to see something dynamic? If it's dynamic, how fast you should image? What is the frequency of acquisition? And at the end, what do you want to observe and quantify? I always suggest people to do a pilot experiment. Don't go there with your experiment saying, oh, I'm going to run my experiment to get my result before my lab meeting. No, doesn't work. Do a pilot experiment. It's less frustrating if it doesn't work, and it doesn't work. It will not work. So in your pilot experiment, choose a very simple sample, something that works all the time, but cells that survive easily, or a tissue that is very bright. Choose the right microscope. If you don't know, come to us. We will guide you towards the best one or the more appropriate one. If you can also test different microscopes just to make sure that they are working for your purpose. And before you get to the microscope, check online with the fpbase.org the compatibility of the fluorophore, the light source, the objective. If you don't want to do that because you don't want to try, or you just don't have time, just come to us. We will do the work for you. Microscopy is all about compromise between resolution, sensitivity, depth, and speed. And we have techniques dedicated for each of them. So there is not a single technique that can do all of them. You have to kind of choose your side, and depending on what you need. You don't need to have a very nice and crisp image to get the data you want to. I show you the example with the spinning disk. Also, plan your positive and negative control. When it doesn't work, it's okay, as long as you know why it doesn't work. If you don't know, then it just doesn't move, it just doesn't go forward. So positive and negative control are the most important things. If you do your pilot experiment, do it until you get the numerical data. Some people just go to the microscope, take some images, they look at the image, oh, it looks good. No, that's not enough. You have to go and kind of analyze, and this will be in session 2 and 3, until you get numerical data, just to have the graph, or just to understand if your picture is good enough to get numerical data. If it doesn't work, then you can actually modify the way you acquire images to get the data you want. Sometimes it's just easier to change the way you do your acquisition to better fit the analysis. It's too hard to have a lot of data, but you cannot analyze. And a bit of methodology. If you choose a way to do things, just keep the same. If you change in the between, it will change your result. Using an objective, I can talk an hour for the objectives, but you need to know how to read an objective. This plan APO, it's about correction. There is a spherical and chromatic aberration in microscope objectives. I didn't describe them, but there is objectives that are better than others for this kind of correction. If you don't use a microscope that is corrected, the focus, for example, the DAPI and the sci-fi will be at different level. So if you take your image, you say, oh, my DAPI is blurry, but I can see clearly my GFP. Why so? It's because the objective is not good. The magnification is actually a very subtle term. What I like it better is, but if you use a low magnification objective, you're going to see a wide field of view. And this is sometimes interesting, but doesn't give you a better resolution. What gives you the resolution is the numerical aperture. They are both linked somehow, but if you want resolution, just keep in mind the numerical aperture. If you want to see a large field, then choose a lower objective. Then there is immersion that you need to use. I told you that it affects the resolution. If you want more resolution, use a oil or silicone-based objective. If you want convenient usage, maybe air objective is better. I told you about contrast method. This is an important value. This is the cover slip thickness. Most objectives are made for 0.17 thickness of objective. So if you don't use that for 0.17, it's going to create blurriness. And finally, the working distance. It's the distance between the objective and the focal plane. So it tells you where it should be your sample. Here it's 4 millimeters, so we're quite far from the sample. It's fine, but sometimes you can get very, very close. Just a reminder about resolution. It depends of the numerical aperture and the wavelength, and it's different for transmitted light and fluorescent. You choose a shorter wavelength, you have a higher resolution. You choose higher NA, you have better resolution. Obviously, the company doesn't make it easy. They just have colors codes, but it's different for all of them. So here it's a blue, here it's a blue, here it's a blue, but it doesn't mean the same or not. We don't really know. But the nomenclature here, it's kind of similar. So read the objectives just to make sure you're using the the good objective. This is an example. Sometimes you have a correcting ring on the objective. This correcting ring is actually useful. I know a lot of people just bypass this thing. It's made sometimes for correcting for the thickness of the cover glass. It should be on 0.17 because most of the cover glass has 0.17. But sometimes people move it. You know, some user before they just move it. If you do that, then you get a blurry image instead of having a clear image. Another thing important about the objective is the transmittance. Not objective transmit the light the same way. So here you have an objective that is very good here between 400 and I would say 600, but it's very bad above 600. So if you want to look at sci-fi, it might be an issue. You have better objective that can be a better transmittance all the way at all the wavelength. There is resources for the objectives, and I put them here. But I think the best objective for the microscope that are the Faculty of Medicine, it's actually the people who are responsible for the microscope that give it to you. So you can go to the company, but the wiki might give you better information. When you go to your experiment, always do a test with a sample test. I usually leave sample slides next to the microscope. Do all the adjustments and the parameters of the software on the slide so it doesn't bleach your sample. You can also restore the parameters from the software if you save your experiment protocols. If you need to do bright field, do a color elimination. And Monique Vasseur did a very, very nice protocol to how to do color elimination. This will improve a lot your transmitted light image. Sometimes it's hard to find the focus. I think this is the most difficult thing when you go to the microscope is, I cannot see anything. It's all blurry, or I don't see anything at all. There is actually step-by-step things you can do to find the focus. One other thing is calibrate the Z. So if it's motorized, you can calibrate, and then you have a number where you can go back to this number and you know it's going to be in focus. You can also check that the cover slips is toward the objective. We don't put the slide upside down. The cover slips always goes toward the objective. And if you use an objective with a long working distance, then you're going to be a safe place because you know that your focus is going to be far enough from your objective, so it's going to be very safe. So use a long working distance objective to find your focus. You can also, if you're using a confocal, open the pinhole so you can see a non-confocal image. On the acquisition, always start with a high wavelength. Always start by the red. Why? Because the red has lower energy. It doesn't bleach the blue and the green. So always start with the red and then go down until you go to the UV. Do your adjustment where on your sample where there is nothing, not like an interesting region. If you go to your region of interest, you're going to bleach your region of interest by doing the settings, and then it's going to be lost. So bleaching is important. So just put the light only when you have to, when you're doing acquisition. Your eyes is also biased towards the red. You see better the red than the blue and the green. So I always advise people on the monitor, on the computer, always put it in gray, because a red light that's showing in gray and a blue light showing in gray, they will appear the same, so you can appreciate them the same way. Do not saturate. And this is saturation. I didn't go into details for lack of time, but if you saturate a pixel, there is no way back. You cannot go back to something that is not saturated. So there is auto exposure features on some systems. Use it because they know better than you sometimes. Save only the images that are useful. I know that you take images and you save, but at the end, if you have a bunch of images that are sometimes big, and you want to sort out which one are useful, it's going to take you a lot of time, and sometimes you just don't want to do it. So just at the microscope, save the ones that are useful, and use the format that is given by the acquisition software. So if you are using Zeiss, use the Zeiss format. If you are using Nikon, use Nikon format, because we are giving you metadata within the file. You can always open them with Fiji and ImageJ afterwards, but save them into the proprietary format so you can go back to the metadata if you need to. If you want, also, you can use the binning. Binning is basically a cooperation on the chip from a camera. So if I have 4 pixels, I do a binning 2 by 2. This 4 pixel will act as 1 pixel, like here. And this is good because it has several advantages, and I'm going to show you in a second. The bad thing is that it decreases the resolution, but sometimes it's a compromise you are able to make. If you do binning, it's going to reduce the size of your file. It's going to reduce the resolution, but it's increasing the sensitivity and also the speed. So knowing binning and using it when you need to, it's actually a good thing. If you save your file, do not use things that are kind of specific characters. My experience with acquisition and saving data and managing a lot of data is to have variables separated by an underscore. If you do that, then you can do a very large scale analysis because your file naming is very consistent. So it doesn't matter, you can choose whatever you want. I put the date first, but you might want to put it at the end. I put an experience and maybe my control or my test, a sample here, and then a random number. But using exactly the same nomenclature will help you a lot with session 3 and session 2. Yeah. There is a lot of different softwares that are available for analysis. I keep a repository of the software that you might need in microscopy onto the wiki. So go back to the wiki. You can click on the link and get directly to this thing. There is also a very nice article that has been published in 2 months ago about the bioimaging analysis software. So this is freely available on bioRxiv. I put some reference about microscopy if you want to learn more. There is the iBiology class that are very well made, but it takes a bit of time to listen to them, especially because most of the time the teacher that gives this class are older, and so they speak very slowly. But the good thing is that you can actually double the speed using YouTube, and you can listen to the 1.5-hour class within 45 minutes. There's other resources that are available here, but this one are not video. I put the link of all the softwares that I know and that might be useful in microscopy here with the link directly to the website where to download them. I also put a software utility box, softwares that you might know of or not, but I found them useful into microscopy world. Most of them are freely available. One other thing I want to raise is GitHub. If you write some part of the code for analysis, put it on GitHub and you will have feedback from people. It's freely, so it's a repository of codes or programming features. It's free. I have one. You can go and pick up whatever I've done before and use them for yourself. It's freely available. Questions? And just to remind you to send me and prepare a data set for session 2 and 3 and send me a sample. And with that, we have 1 minute for questions. Whew. I made it. Ça peut être une image, ça peut être plusieurs images, ça peut être une vidéo, ça peut être quelque chose qui te sert dans tes études, dans ta recherche. Si tu n'en as pas, tu peux aussi me parler d'un sujet, puis moi je vais aller chercher des données qui ressemblent à ça. Mais dis-moi ce que tu veux faire dans ton labo en recherche, et moi je vais peut-être trouver des données. Si tu, par exemple, tu me dis, moi ce que j'aime bien, c'est calculer la fréquence de division des bactéries. Je vais aller chercher des bactéries qui se divisent et on va faire des analyses pour compter le nombre de bactéries par image, par exemple. Any question on Zoom? Non? Alors, vous êtes libre.